Hi there, this is Cell and Gene TV. We're at the Cell Therapy Manufacturing and Gene Therapy Congress, and I'm Claudette Hodge, and I'm here with Behnam Ahmadian, who is Global Head of Process Development of Cell and Gene Technologies at Lonza Pharma and Biotech. So hi there, Behnam.
Hi.
Nice to meet you.
Nice to meet you.
So earlier you were talking about the manufacturing of cell and gene therapies, and you did touch generally on allogeneic therapies. So within cell and gene therapies as a whole, what are the biggest differences and similarities in scaling of manufacturing processes, especially for allogeneic?
Yeah, so if you look at the cell and gene therapy applications and manufacturing processes, specifically for allogeneic applications, the manufacturing process can be divided into multiple unit operations. These unit operations usually start from, the starting material, processing of the cells and isolation of cells, to expansion, depending on the application, differentiation, then downstream processing and characterization. So some common terms and unit operations are applicable to all these unit operations, from cell isolation, expansion, downstream processing, and also requirements for characterization. If we look at these processes, though, for each application and depending on the characteristics of the cells, the cell types, the requirements from manufacturing perspective and GMP manufacturing perspective in particular could be different. The cellular characteristics could be different. Some of the cell types are anchorage independent cells, and some of the cells are anchorage dependent cells, and therefore the way these cells are growing in the culture could be different. Some of these cells might be attaching, requiring a surface to attach and grow. Some cells need to stay in contact to maintain the cellular microenvironment niche and grow in the form of aggregates. So the cellular characteristics play a role in terms of which unit operation we need to use. The type of application and phase of application can also impact how we manufacture these cells, what type of scaling strategy we use for these cell types. In particular, which type of bioreactors and mode of culture we need to use. Do we need to use, for instance, microcarrier-based technology for growth expansion of the cells? Can we use the cells that are growing and forming aggregates in suspension bioreactor? Also, from in-process control and process monitoring perspective, different type of analytics could be applicable for different applications. For instance, when it comes to processes that are involved with directed differentiation of pluripotent stem cells into a specialized cells, what we need to focus on is not only expansion and proliferation, but also phenotypical change and directed differentiation. So the type of in-process control, in-process monitoring that we apply in the scale-up process could be different. We require more stringent sort of in-process monitoring to make sure that the cells are not only expanding, but when needed during directed differentiation, they can differentiate from pluripotent stem cells into multipotent, and then into progenitor cells, and finally a specialized set. Also, the final product characterization and testing requirements for different cell types could be different. And the strategy that we take from release assays or characterization assays could be different depending on what is available. So in a sense, I would say that yes, there are a lot of similarities from large high-level unit operations, but there are major differences when it comes to the cellular requirements, the phase of application, how we use these cells for different cell therapy applications.
So what would you say are the biggest process development challenges for allogeneic cell therapies, and also what is Lonza doing to address the challenges?
So some of the major challenges in the field of cell and gene therapies are essentially the type of unit operations that are currently being used in manufacturing processes. These unit operations are usually open manual 2D unit operations. Also, I would argue that there are significant lack of analytics, process analytics, in process control and in process monitoring techniques and methods that can be used in development and also manufacturing of the cell and gene therapies. The technology development is certainly behind the pace of growth for cell and gene therapy applications. Even the bioreactor technologies that are being used currently for the cell and gene therapy applications have been previously established for biological applications, and they are certainly lacking some of the key features that we would want to focus for cell and gene therapy applications. So from my perspective, from process development and also analytical method development perspective, I would like to focus on automated computer-controlled bioreactor systems. But in order to do that, I would need to have access to the technologies and method that can translate the current processes from open 2D manual unit operations into more computer-controlled bioreactor. I would like to have access to analytical methods and technologies that can be used in the control of the process, and also the type of bioreactor systems should be applicable for the specific application, cell and gene therapy application, and the type of the cells that we want to use in the manufacturing process. Last not least, when it comes to translating and moving the cells into more automated closed system, some of the major focuses has been recently on type of bioreactor that is being used, but I would like to say that after expansion and or differentiation of the cells for specific applications, we would need to make sure that appropriate downstream processing technology is available. And this is area that requires further development, and I'm hoping that the downstream processing technologies will evolve and will allow us to move towards more automated computer-controlled closed system.
So those issues, speaking about those, as you just said, have there been any developments in either case of allogeneic or autologous? Anything like equipment design or analytical test methods or any other technologies?
Yeah, so we know that the pace of growth for cell and gene therapy applications is really, really fast. We see a lot of applications, and we also know that probably the pace of growth for this technology development is not in sync with the cell and gene therapy applications. But I'm very optimistic and hopeful. I've seen significant developments recently. For instance, when it comes to downstream processing, I've seen some of the new technologies, acoustic cell separation, for instance, or some of the technologies that are being used for autologous applications, for instance, LOVO system, CPAC system, they are being used more rigorously in the manufacturing processes. We're certainly moving from the open manual washing and cell processing systems into more closed, programmed and controlled cell processing systems. Also, I would like to mention that for us it's important to see more platform technologies that can be used in manufacturing of cell and gene therapy applications. Some advancements in this area are the iPSC manufacturing platform. Actually, we have been working at Lonza on the cell culture system and establishing the iPSC manufacturing platform, and now we are helping our clients to use this manufacturing platform to take care of important aspect of allogeneic application when it comes to iPSCs, generating high quality GMP compliant iPSCs upfront and using the platform that allows our clients to generate the iPSCs, and then these iPSCs still maintain the quality and the potentials to be used in directed differentiation. Therefore, this allows from timing perspective for our clients to get them where they want to be from clinical and commercial manufacturing perspective. I would like to also highlight on the importance of analytical methods. Assays and analytical methods are probably one area that has been somewhat overlooked when it comes to cell and gene therapy applications. unfortunately, most of the focus on manufacturing is the process. But from a process development perspective, I cannot have a optimized, robust, and reproducible manufacturing process without having proper tools and analytical methods. Therefore, I need to have access to the tools and analytical methods that are reliable. And this has been evolving a story. We know that new flow cytometry techniques are evolving. We know that the ddPCR, for example, methods and techniques are evolving, and we can use on both cell therapy and gene therapy applications using analytical methods, addressing some of the key challenges that we've seen in terms of establishing the process that is robust and reproducible. And also is important to highlight the importance of some of the new characterization methods, whole genome sequencing, for instance, gene array analysis methods, that maybe we can use them as characterization tools now to evaluate and understand the quality of our starting material or the cells when they are changing during the manufacturing process into more specialized cells. Hopefully these methods will be more reliable, and we can qualify and validate these methods in the future to essentially take care of all the critical quality attributes of the final product. All in all, I would like to say that technologies are evolving. I would like to see a little bit more in terms of speed and pace of growth, but we remain hopeful that we can move towards more industrialized manufacturing using more high-level cell and gene therapy technologies. Focus on efficiency also, using high throughput technologies for cell therapy applications as well.
So the analytical methods, the technologies, and the manufacturing platforms that you mentioned there, are there any that are specialized for cell and gene therapies?
There are some equipment and technologies that are being specialized for cell and gene therapies. We know that at Lonza, for example, we focus significantly on automated closed system. We have been working on an automated closed system for autologous application we call Cocoon system. This is an automated closed system right now focusing on autologous applications. And this is a technology that we are hoping that we can help us to commercialize and industrialize the autologous applications. We are also working with technology developers to take some of the assays and analytical methods in the manufacturing processes. Here we work on some of the analytical methods such as flow cytometry, PCR-based, molecular biology-based methods, to not only use them for in-process monitoring, in-process control, but also qualify them, validate them to use more for the release of the product. Obviously, we would like to establish that the basic relationship between critical quality attributes of the product with the critical process materials and critical process parameters that are being used in the process. And then we are hoping to use these methods for the final characterization and release of the final product.
Okay, so on to autologous therapies. What's Lonza's approach towards automation of autologous therapies?
Yeah, as I mentioned, we've been heavily focusing on moving away from open manual 2D unit operations. When it comes to automation, there are two approaches that one could take. One is modular approach for automation, meaning that you replace open unit operations with the automated unit operations, and also one could take end-to-end automation approach. Of course, when it comes to end-to-end automation, we need to make sure that technology and equipment that allows us to have end-to-end computer control automated system is available. And this is a developing area that the technology developers are working on. Lonza is also working on using Cocoon and developing Cocoon into more end-to-end approach for autologous applications. But currently we are also heavily focused on addressing our clients' need during the process development and moving the open manual 2D unit operations to more automated system. For instance, when it comes to autologous applications, for the isolation step, we focus on moving away from the manual phycal separation type of unit operations into more automated system where we use bead technologies, selective bead technologies for isolation. Washing and cell processing steps, we move away from centrifugation and manual steps to more automated systems using the LOVO system, for instance, CPAC system, really high-tech technologies. And I could imagine that honestly the manufacturing platforms and the technologies can play a significant role. I want to argue that the manufacturing process are going to move into more high-tech type of manufacturing manufacturing processes that use the high-tech technologies for different unit operations. That is a developing story, and we are certainly moving towards allowing and enabling the use of innovative technologies in manufacturing setting. A major focus for autologous applications for us is assay development and use of analytical methods. We have done extensive work when it comes to using different type of assays for evaluation of the different unit operations, starting from cell isolation to washing and processing, activation, gene modification, evaluation of the transfection or transduction efficiencies, depending on the way that we use for gene modification, the method we use for gene modification, as well as downstream processing and characterization. We've been focusing significantly on using reliable assays and methods to evaluate the cells from starting materials to intermediate materials and then final product.
Great. Well, as just one last question, as a whole, for cell and gene therapies, if you're fast forwarding to the year 2030, how do you see the industry changing over the next 10 years?
Yeah, I think the industry for cell and gene therapies will change and move towards more automated, computer-controlled closed systems. I'm hoping that we, Lonza, as CDMO, will play a significant role in that. We would like to move away from unit operations that could create quality, quantity, and also maybe efficiency concerns for manufacturing processes. As the technology grows and new technologies are evolving, new methods and assays are evolving, I'm hoping that we'll move away from open manual 2D unit operations into 3D computer controlled automated closed system. I'm hoping that in the next 10 years, the analytical methods in process control, in process monitoring methods can be used. PAT, process analytics, is going to be significant focus, and we will be moving into technologies that are more established, more standardized approach when it comes to the whole services from development to manufacturing and moving from clinical to commercial manufacturing. We'll move towards the technologies that allows us to work toward the commercialized ready manufacturing applications. I believe that CDMOs can play a significant role in this with developing or improving platform technologies. Currently, we don't have many probably in the industry, but these platform technologies will evolve for different cell and gene therapy applications. CDMOs can play a significant role in development of these platform technologies, implementing platform technologies in manufacturing settings, and also addressing both timeline and quality challenges that we might see right now. And I'm hoping that this industry will grow the same way that the biologics industry grew several years ago. And the pace of growth will depend on the successful clinical applications, obviously technology development. We know that a lot of folks are active in the industry, and we, Lonza, as a CDMO, we hope that we can play also an important role to take these manufacturing processes into the next level, into more industrialized setting.
Great, that's really great. It's been lovely speaking to you, Behnam. Thank you very much.
Thank you very much.